Good afternoon, everyone. Thank you for joining for our presentation. Today, I'm going to share with you how we are developing curative and affordable cell therapies that will eliminate blood diseases from the planet Earth. When we formed Garuda 3.5 years ago, it was based on 4 platform pillars. First platform pillar was about our unique ability to develop off-the-shelf induced pluripotent stem cells, aka iPSCs. The unique thing about our technology is that we are developing a set of lines that will allow us to perform 12 out of 12 HLA compatibility while maintaining only 15 lines that will cover half of the world's population. As you know, iPSCs can be used to convert any cell type in the body. In our case, we use iPSCs to make our second platform pillar, which is off-the-shelf hematopoietic stem cell. I'll be spending a lot of time on a HSC platform later, so I'm going to skip the introduction on it. But we also have a unique ability to develop off-the-shelf gene-empowered hematopoietic stem cell. I'm sure many of you know HIV/AIDS have been cured at least in 5 patients, but that could never be democratized beyond those 5 patients because nobody in the world knows how to perform 100% homozygous safe editing of hematopoietic stem cell. And the fact Garuda used iPSC as a starting material, that puts us in a unique position to perform those 100% homozygous editing at the iPSC stage, make sure that genomic integrity is preserved, and then we expand the clone and differentiate them into pre-edited transgene-free gene-empowered hematopoietic stem cell. So examples on a slide are just the beginning, but it just allows us to expand beyond those disease areas. And the fourth platform pillar is our unique ability to develop off-the-shelf blood cells. As we all know, there is elephant in a room that off-the-shelf blood cells don't work, which is true, because we start traditionally from something called CD34 positive cells without knowing those CD34 positive cells are not hematopoietic stem cells. So if you start from material which doesn't make sense, what comes out in the end is just not functional or bioequivalent what we've been promising to our investors and patients. In our case, since we have made engraftable hematopoietic stem cell, it just puts us in a position of power to develop bioequivalent functional durable hematopoietic cells like off-the-shelf myeloid cells that can undergo phagocytosis, off-the-shelf platelets that can actually clot, and off-the-shelf NK cell, T cell, B cell, and progenitor T cell. Obviously, no company on planet Earth would have opportunity or money to pursue all of this. So we have decided to prioritize 3 first-in-class and best-in-class product opportunities. Our first-in-class off-the-shelf hematopoietic stem cell would have an opportunity to potentially treat 120 plus diseases. We just received very positive and productive regulatory feedback from both EMA and FDA, and we foresee ourselves opening CTA and IND very soon to dose a patient hopefully by the next meeting on MESA. Our second first-in-class and best-in-class product is our ability to develop off-the-shelf CCR5 deleted hematopoietic stem cell to provide hopefully curative therapy to AIDS patient in outpatient setting. And our third off-the-shelf first-in-class product opportunity is our ability to make off-the-shelf progenitor T cell. I know we have heard many talks around people trying to make alpha beta T cell, gamma delta T cell, T memory cell, T effector cell, T regulatory cell, NK cell. I can go on and on and on. But I think we should take a step back and ask ourselves why Mother Nature made all of these cells together and not one at a time. So what's happening is when we are trying to make alpha beta T cell, we miss out on gamma delta T cell. And when you make gamma delta T cell, you miss out on something else. So we thought about this for a long period of time, and we decided that instead of making one cell at a time, let's just make a mothership cell, which is progenitor T cell, and let the Mother Nature do the magic, where this cell would engraft in thymus and would make the cell the body need at a time. And this will provide durable, off-the-shelf, long-term supply of immuno-oncology or immune disorder-based cell therapy, which you've been waiting for forever. So that's where we are in our development. The fact we can make off-the-shelf hematopoietic stem cell, it comes with a bunch of bells and whistles, which I would like to walk you through. Our product is available off the shelf and it's 12 out of 12 HLA compatible. So the beauty of this technology is nobody would have to look for a bone marrow match again. You go to hospital and you have something compatible to you immediately available. And I'm sure you know, patients who are biracial, they have zero chance to find donor. And there are many patients out there who just don't find donor for a long period of time. And if someone has leukemia, it's not going to stop spreading while we are looking to find donor. So having something available off the shelf has its own value. The second major advantage of our technology is that scale is not an issue. So we can pursue something called mega dose, which means higher the dose, faster the chimerism, sooner the patient can go home. And as you know, cord blood transplant have fallen off planet Earth because it takes forever to engraft. And we can't just put patient for 4 months in ICU under immunosuppression. The third major advantage we have is that our cells actually engraft in absence of conditioning. So diseases like thalassemia, sickle cell, bone marrow failure, immune disorders, where conditioning can be avoided, should be avoided, and we have an angle to approach that. The fourth advantage we have is our product does not have T cell. So if your graft doesn't have T cell, there cannot be GVHD. And this pretty much transforms how we think about hematopoietic stem cell-based transplant moving forward. The fifth advantage we have is that our product is a pure cell therapy. This is neither gene therapy nor cell and gene therapy. So the cell which actually enters in patient body has no virus, no vector, no episome, no gene editing material, or no free-flowing Caesar, which might do whatever thing which we cannot imagine. So this really elevates the safety profile of the product we are building here, and that puts us in a very unique position in discussing our product with regulators. And most importantly to me, we want to make sure we develop a product which is accessible to patients across the globe and not just for United States of America because reimbursement is very high. So we have from the beginning kept costs so low, and that's a fight I have with my manufacturing organization every day. Whatever we do, the cost has to be low so people in Asia can afford it. That's where half of the world is. If they can't afford it, whatever we do here, it just doesn't make any sense. So having this sort of opportunities put together, we felt we are uniquely positioned to build one product that will cure 120 diseases. And that's not our statement. This is based on 50 plus years of experience we have in the field of bone marrow transplant, where we have used it to cure 120 diseases in 1 million patients. And no such cell therapy out there who has enjoyed such a robust potential. So before I go too far, just want to clarify what hematopoietic stem cell is and what hematopoietic stem cell is not. So our approach is our ability to make self-renewing long-term hematopoietic stem cell. But people have forgotten that in Mother Nature, hematopoietic stem cell asymmetrically divide into one progenitor and another hematopoietic stem cell. And it's progenitors who actually reconstitute multilineage blood and not hematopoietic stem cell. But hematopoietic stem cells are the one who provide self-renewing durability. But when it comes to clinical practice, we go by something called 2 million CD34 positive cells per kilo of patient's body weight, but we've forgotten that CD34 does not mean they are hematopoietic stem cell. It can be progenitor cell, it can be endothelial cells, can be hematopoietic stem cell. And if you do limiting dilution assay, you would know only 1 in 10,000 of the CD34 positive cells are true hematopoietic stem cell. So often the time we don't know what we're putting in patient's body. And that's why we have graft failure at the moment you gene edit hematopoietic stem cell. So the way we figured out this technology was more around, we have a very rebellious mindset. We decided to pretty much challenge the dogma in the field that hematopoietic stem cell transplant being called as bone marrow transplant because people thought they come from bone marrow, but that's not true. When I was a professor at Harvard, my lab, and around the same time, a few others demonstrated that hematopoietic stem cells are actually born in a tissue called aorta gonad mesonephros region, which is like a blood vessel-like structure. And once they're born here, they migrate to fetal liver for expansion. And once they migrate to fetal liver for expansion, then sufficient numbers of cells are being produced, they go to bone marrow to sleep and not to make more of them. And once they sleep, one of them would wake up and differentiate and would go through asymmetric division to make blood as we need. So our technology was more around trying to understand how Mother Nature makes this cell and try to recreate that whole process ex vivo in a dish, which is obviously there is no electropoietin in a body, right? So it's pretty much straightforward natural processes. So long story short, after figuring out how to make hematopoietic stem cell, we also learned that making hematopoietic stem cell was a holy grail kind of discovery, but making it available to rest of the world is a bigger headache we have to go through. And as you know, we can't do HLA null universal approach, because if you do so, there are three major issues in our case or any other case. One is you lose the immune surveillance capacity, and cells would acquire mutation and body will never detect it. Second is you lose don't eat me signal, and to overcome this issue, we have to over-engineer cell by putting HLA-E, CD47, and whatnot. And third is this HSCs would actually make T cell. And if T cells don't have MHC presentation capacity, it's just a waste of the approach. So we kind of went through organic way of evaluating situation, and we came up with this unique proprietary formula, as I mentioned before, which allow us to generate 15 lines that will cover half of the world's population. At the same time, it will allow us to have 12 out of 12 HLA compatibility. So we use this line to develop off-the-shelf bank of hematopoietic stem cell. So anyone is looking to find bone marrow donor, instead of going around waiting forever, you just go to hospital, you have something available for you and ready to go home. So this whole approach will look very similar to ABO blood banking approach, and that's where the power is here. So as far as Garuda is concerned, we are at a point where we are able to manufacture a batch which will allow us to support our phase 1, phase 2 trial. And as I mentioned before, we had to make sure that we know what's the end game here. And as you know, I'm sure many of you have interacted with investors recently. Everybody hates about cell therapy, the fact that if it works in science, doesn't work in manufacturing, works in manufacturing, doesn't work in clinic, works in clinic, there is no regulatory clarity, and if it works in regulator's mind, there is no commercial path. So we make sure that we check all of those boxes from the beginning, and we convince our stakeholders that we know what we are doing. And we put all of these factors together, and we decided to prioritize 2 diseases in parallel. So we'll be pursuing a transfusion-dependent thalassemia at 5 sites in Italy, and in parallel, we'll be pursuing, a basket trial in bone marrow failure syndrome at 2 sites in Italy, 3 sites in France, 3 sites in Brazil, and 2 sites in the United States. And the beauty about this approach is that it's the same product. We are just repurposing from one disease to another. So when we have to go from second to third disease, all we have to do is update the clinical protocol. And that really keeps the cost of development very low. So in the last 1 minute, I hope I have convinced you that we are able to transform the field of hematopoietic stem cell transplant by walking away from current dogma, which is, unfortunately, one patient, one product, one disease at a time, and we hope we can fix this by offering something called off-the-shelf hematopoietic stem cell, which will be considered as a one product for 120 plus diseases with all the bells and whistles you see on this slide. So with that, I would like to thank you for attention. Happy to take any question you might have in the last 16 seconds. Great, thank you.